It's great to see everyone here. We work together as a team. My name is Guang. I'm going to report a new discovery. We found a new spotted fever group, Rickettsia, in rabbit tick, Haemaphysalis, leporispalustris in Maine. So we all know Rickettsia can transmit disease to human or animals during the blood feeding by infected ticks, mice, lice, or fleas. Based on the phylogenetic analysis, they are at least four groups. The largest group is spotted fever group. Within those group, there's the most lethal disease, Rocky Mountain spotted fever, caused by the Rickettsia. This is most lethal tick-borne disease in the United States. So I think up to today, there are 5 Rickettsia groups found in the United States. Rickettsia parkii, usually we found this in Southeast United States, but in this year, Rickettsia parkii and its vector tick, Gulf Coast ticks, moved to Connecticut. So I think we'll find this next year probably in Massachusetts. Rocky Mountain tick fever was transmitted by, usually we think it's transmitted by dermocenter ticks, but recent study shows brown dog tick is probably the most important vectors compared to dermocenters. Cape Cod used to be the hot spot for Rocky Mountain spotted fever, but it's very, very rare in New England now. Most cases found in the middle of the United States. The last 2, Pacific Coast tick fever was transmitted by a different tick species only found in California. And last month, a new Rickettsia species, CA6269, they proposed a new name called Rickettsia lenii, was reported in the EID journal. It was transmitted by rabbit ticks. So we test a lot of ticks. In those 2 years, we test over 6,000 each ticks. Here is our workflow. So once we receive the tick, we first ID it using the morphology characters. We divide it in 2 groups, exotic tick and non-exotic ticks. So we use exotic package, test exotic ticks, and here is the example for non-exotic ticks. For each tick, we test 3 different packages. The first package contains all the species tests. The second one tests, we call it NIT1, tests Rickettsia borrelia. The last one, we test tularemia, anaplasma, and the Rickettsia. So each ticks, we run 3 tests. So last year, we received 3 ticks. At the beginning, we received 3 ticks from Maine. It was kind of surprised. We found 1 Rickettsia positive, Rickettsia positive. It's really a surprise because we test, since 2006, we test over maybe more than 80,000 ticks. We didn't even find 1 Rickettsia positive. So this was our first Rickettsia positive. We report this to Ali. Then Ali sent another 190 ticks. I believe 10 of them we test were positive. This year, she submitted more rabbit ticks. In total, you can see this number, 18 of 298 rabbit ticks collected from Maine positive. The rate is pretty high. It's 6% of them are positive. I have to say, why Maine group collects those many rabbit ticks, I don't know. So we got a small number from other states. That's a lot of rabbit tick from Maine. So once we test the positive, we want to know what kind of species, because Rickettsia is very, very similar. The sequence is very, very similar. Although this assay say that's a CDC assay, says that's Rickettsia. We want to confirm it. We use the multi-local sequence typing method, MLST method. We use 4 genes, outer membrane protein A, B, and the surface cell antigen 4 and GAL-A. We PCR those 4 genes, purified, sequence that, then align with available genome sequence. We build a phylogenetic tree and found the top one, the right color is the Rickettsia we found from Maine. What do we know from this tree? The first we know this is the new Rickettsia. And we know probably this Rickettsia will cause some kind of human disease, because you can see it grouped together with the Rocky Mountain spotted fever pathogen Rickettsia and 2 human pathogen from California. So it's very likely that's pathogenic. And we know it can transmit from an adult female to next generation, because we found this pathogen in larval, nymph, and adult, all the stages. It's also more questions. We need more studies. First is, will this kind of tick transmit this new Rickettsia to human? I guess yes, because this tick is a 3-host tick. It's mainly fed on rabbit, but it seldomly fed on birds and small animals. It does feed on human, but very, very rare case. So the recent paper found, okay, 2 patients, they encounter rabbit ticks, but the frequency is very low. In 20 years, they have 2 patients. This very interesting thing is, 1969, 13 Rickettsia Rocky Mountain spotted fever case was reported from Cape Cod. But if you look at the time, it's from April to June. They thought this was transmitted by dog ticks. It's very hard to find dog ticks at this time. And from this 55 years, nobody even isolated or detect Rickettsia from stomach centers. So maybe, I guess, that's a missing link here. So we need to do more study for the rabbit tick and this new Rickettsia. We don't know, right now we don't know the host. I think Leslie told me this morning, all the ticks we tested all the flag ticks. We don't know the host. Definitely we need more studies. This is teamwork. I want to thank for the Maine group and the UMass group. And Steve, thank you, Steve.